Now we will discuss pre-mRNA processing. As you recall, in bacteria, transcription and translation take place simultaneously. While the 3' end of mRNA is undergoing transcription, ribosomes start to attach to the Shine-Dalgarno sequence near the 5' end and begin translation. This is very typical for bacteria. However, in eukaryote, you recall, nucleus is separated from cytoplasm by nuclear membrane. Transcription takes place in the nucleus, while ribosomes, where synthesis takes place, are in cytoplasm. This actually allows for additional level of gene expression controls at the level of processing of pre-mRNA. Eukaryotic cells process extensively mRNA after transcription. First change which is taking place is addition of 5' cap. Addition of cap to 5' end of the pre-mRNA. This facilitates binding to ribosomes as well as it does increase stability of mRNA. Afterwards, there is 3' end modification. At 3' end, we can find cleavage site as a consensus sequence where pre-mRNA is cleaved and poly-A tail is added. You remember poly-A tail is not encoded in DNA template. This increases stability of mRNA and also increase binding of mRNA to ribosomes. Afterwards, RNA splicing takes place. During RNA splicing, non-coding regions, introns, are removed from pre-mRNA, and this actually is required for export of mRNA from nucleus to cytoplasm. Splicing also allows to produce more than one protein from the same template, from the same coding sequence, and it is possible due to alternative splicing. Now the last modification we will discuss is RNA editing. While all previous modifications take place on pre-mRNA, RNA editing takes place on the mature mRNA. During RNA editing, nucleotide sequences are altered already on form mRNA. The addition of the 5' cap takes place rapidly after the initiation of transcription, and it is a very unique 5' bond. As you can see right here, 7-methylguanine right here is attached through 5' bond to 5' of the RNA. It is this very particular and unique bond is carried out by enzyme that associates with RNA polymerase II. So basically, ribosomal RNA, tRNA, and small nuclear RNAs do not associate with this enzyme, and therefore they are not capped. So what is the function of 5' cap? As we mentioned before, there are cap binding proteins which recognize cap in the cytoplasm, and this cap binding protein bring mRNA into ribosomes. They mediate attachment of mRNA to ribosomes. So 5' cap is very important for mRNA binding to ribosome for initiation of the translation. It also increases stability of mRNA. The second pre-mRNA modification is the addition of poly-A tail. Approximately from 50 to 250 adenine nucleotides are added to the 3' end of the pre-mRNA. Now, these nucleotides are not encoded in the DNA. So how these nucleotides are added? We know that RNA polymerase II synthesizes or transcribes well beyond the end of the coding sequence. And within that sequence, we can find so-called polyadenylation signals. One of them is represented by consensus sequence AAU-AAA, which is upstream of the cleavage site. Another one is downstream of the cleavage site, and it's U-rich sequence. The sequences are recognized by proteins which binds and bring upon the cleavage right on the cleavage side. Afterwards, the enzyme associated with the polymerase adds nucleotide adenines to the 3' end of the cleaved mRNA and actually brings upon synthesis of poly-A tail. So what is the function? Poly-A tail increases stability of mRNA, and it is due to the proteins which binds to this poly-A tail and protect mRNA from digestion. It also facilitates attachment to the ribosome of mRNA. So it's not only stability, which is a primary function, but also it facilitates attachment of mRNA to ribosomes. The other major type of modification of eukaryotic pre-mRNA is RNA splicing. RNA splicing refers to the removal of non-coding sequences, which we call introns. So to work properly, splicing requires presence of 3 consensus sequences. It is 5' consensus sequence and 3' consensus sequence, which both contain splice site where pre-mRNA is cleaved. Third consensus sequence is branch point. Branch point is formed by adenine. It's critical for proper splicing. If you mutate adenine in branch point, splicing will not take place. Also, the sequence surrounding the branch point is not, it's very variable, it's highly variable. Now, what mediates splicing are so-called spliceosomes. Spliceosomes are formed by 5 small nuclear RNAs and almost 300 proteins. So the spliceosomes are large and highly complex molecular structure, and the composition may vary from cell to cell. So those proteins can be different in different cells, which enables different splicing of the same pre-mRNA in different cells. So how does splicing proceeds? It usually proceeds in 2 steps. In the first step, the 5' of the introns is cut, and 5' cutting actually releases exon 1 from intron. The 5' end of the intron attaches to the branch point to adenine via transesterification reactions. Within these reactions, the guanine, you remember guanine is within consensus sequence of the 5' consensus site, splice site, will interact with adenine so that the 5' phosphate group of guanine nucleotide now attaches to 2' hydroxyl group of the adenine. This attachment triggers second step, which is 3' splicing. As soon as 3' of the intron is spliced, exon 1 and exon 2 are connected. Now the cut out intron or splice intron for so-called lariat structure, which eventually linearized and is digested, broken, and degraded by the RNases within the cells. So, splicing of nuclear introns in pre-mRNA requires 2-step process. As we discussed before, splicing of pre-mRNA takes place within a large, complex called spliceosome. Spliceosome is composed of 5 small nuclear RNA, U1, U2, U4, U5, and U6. U3 is missing. And almost 300 proteins. What happened first is that U1 and U2 recognize consensus sequences in pre-mRNA. U1 recognizes 5' splice site and U2 recognizes branch point. This triggers further formation of the complexes of U4, U5, and U6 together with some conformational changes, releasing some of these small nuclear RNAs, formation of large spliceosome complex, and cleavage of the pre-mRNA at 5 and 3' splice site. Now, what holds spliceosome together with pre-mRNA is base pairing between small nuclear RNAs and mRNA. Now, upon cleavage, the exons are joined together and the introns are released and as a lariat structure. You remember we discussed that before, and eventually degraded. Intron removal processing as well as transcription take place at the same time in nucleus. And in most RNAs, mature mRNA is produced from a single pre-mRNA. However, in certain organisms, such as trypanosomes, the final mature mRNA can be produced by splicing together sequences from 2 or more different RNA, and we call this event trans-splicing. Now, importance of splicing in normal functioning of the cells, eukaryotic cells, is supported by finding that many human disorders, genetic diseases, are caused by mutation which affect pre-mRNA splicing. So, normal RNA pre-mRNA splicing is required for normal functioning of eukaryotic cell. Some introns are self-splicing, so they possess the ability to remove themselves from pre-mRNA. We talk about two major groups, group 1 self-splicing introns and group 2 self-splicing introns. Group 1 introns are found in RNA genes in some protists and fungi. Group 2 introns are in mitochondrial genes, some of them, and they also have ability to self-splicing. So both groups are self-splicing and both form into very characteristic secondary structure. You have the secondary structure of group 1 introns here and group 2. So there are differences in the secondary structure. However, what is the major difference is that group 2 introns, the mechanism by which they self-splice is more similar to spliceosomal mechanism of intron removal, which suggests that nuclear pre-mRNA introns are evolutionarily related to group 2 introns. So group 2 introns form similar lariat structure as do spliceosomes.